Assalamualaikum and good day everyone. So this is a BWJ 32003 Plant Physiology, and today we're going to learn on chapter 3, the first subtopic, which is the cellular basis of plant growth and development. So before that, let's have a look at the definition of the plant growth. So plant growth is an irreversible change in the size of a cell, organ, or whole organisms. Yeah. So there are a few keywords or key points in this definition of the plant growth. The first one is the irreversible change. Okay. So when the plant grows, so they can't go back to being the state where they were before. Okay, so it's the same for the adult as well. It's the same for human as well. When we grow up to adult, we cannot go back to being a kid, a child. Okay, and then what do they grow? They grow in the size of the cell, then they grow the organ as well, and subsequently is the whole organism. Meanwhile, for differentiation is the cells taking on the specialized form and function. And so this is how the cells grow up. Plant development is the orderly and progressive change from seed germination through juvenility, maturity, flowering, and fruiting. So if you realize here, the juvenile, the maturity, the flowering, and fruiting, so this is the plant life cycle. So for this part, so we're going to have a look at the plant life cycle, and we're going to see each step or each life cycle, how it is done or being conducted on the cellular and the molecular basis. So this is the plant life cycle. So we are going to learn on the 5 steps on how is a seed formed. Then once the seed is formed, then how does that seed become a seedling? Then once it is a seedling, it is in a seedling state, then how is the shoot formed? Once the shoot is formed, then how is the root formed? And once they are done with that, then let's see how they flower. So the first one is how is a seed formed. So before that, we have to know the gametophyte of the male and female, because it involves with the reproduction and also the fertilization. So it's the same with human as well. The plants need male and female sources. Okay, so for plants, the male sources is the gametophyte. It comes from the anther, and the gamete is from the 2 sperm cells. And the 2 sperm cells of the male in plants, they are located in the pollen grain or the pollen tube. So for female, the gametophyte is the embryo sac, and the gamete is the egg. So gametophyte ni is the sexual phase lah, and the gamete tu is the reproductive cells. So as we can see, the male, the reproductive cell tu is the sperm cell. Okay, kalau for female, the reproductive cell tu is the egg. So it's kind of the same with the human as well, but it's just similar, but it's not the same. Okay. So in order for the plants to form the seeds, so first they have to go through the fertilization process. So this is the structure on where the fertilization process are happening. So it is when the sperm cells, it is stick in the stigma, and then it will travel through the pollen tube, and it will go into the ovule or the embryo sac, and this is where the fertilization occurs. So once the egg is fertilized by the sperm, then the embryo will develop within the sac, okay, within the female gametophyte, the embryo sac, and this ovule will later become a seed. So this is the simplified structure of a mature seed, and it has 3 basic structure or components. So the first one is the seed coat. Yeah? So the function of the seed coat is to protect everything inside the seed coat. The second structure is the embryo. Yeah, so the embryo is a tiny plant inside the seed. Okay, but before that, I would like to mention that seed coat ni is a dead tissue. So it is a type of tissue, but it's just that it is a dead tissue. Right? So one of the reasons of the seed coat ni is to protect whatever inside the seed. Okay, so embryo ni is the tiny plant. Okay, so it's the baby plant. So it's the same with human. The embryo ni inside the womb of the mother is like a baby human, baby lah. Okay, and the third structure is the endosperm. So endosperm is a type of tissue as well. It's the same with the seed coat. The function of the endosperm, it provides the energy, it provides the nutrition, the nutrient for the seed germination and also for the early seedling growth. Okay, so it gives all the everything, whatever the embryo needs for it to go through the germination process. So that is how the seeds is formed. So let's have a look at the second step. Once the seed is formed, how does it become a seedling? The seeds become the seedling through the germination process. And for the germination process to take place in plants, it has several factors that contribute to the germination process. So the first event in the germination, they must have some water. So there is a water uptake process, and these figures, it shows the difference between the dry seeds and also the imbibe seeds. Yeah? So imbibe ni, it says ni because it is the result of the imbibition process. So imbibition process ni is what is it? It is the absorption of water by the seeds. So that's why we can see the difference in the size between the dry seeds and the imbibe seeds. Because when the plants take up the water and it goes through the seeds, so it results in the rehydration of the seeds, and also it will result in the cell expansion. So that's why we see that the seeds of the imbibe seeds is bigger than the dried seeds. So the seeds, imbibe seeds, whenever we see the imbibe seeds, it means that it has gone through the imbibition process. So it means that they have absorbed some amount of water and they are experiencing some swelling inside their cell, the seed cell. So that's why they are bigger. Okay, and once the imbibition started, the rate of the respiration inside the seed too will increase, and a lot of metabolic processes will resume from the dormancy. Okay, so for seeds they will go through dormancy punya process. Dormancy tu is a process where they will release their water content up to 5% sahaja water content dekat dalam in their seeds. Because dormancy tu, it means that diorang duduk diam-diam, they haven't received the signals or they haven't received the cues yet for them to grow into a seedlings, kan? So that's why they will lose all their water content and they are in the dehydrated mode. So when they have received the cues or the signals, when there is enough water, then they will take up the water, and then it will result in the hydration and also the cell expansion. So this figure, it shows on the water relations and also the seed germination. Okay, so there are three treatments here. On the high water availability, medium water availability, and also the low water availability. So this experiment, it shows that for the seeds that receive the high water availability, meaning that the amount of the water is a lot, rather in comparison to the medium and also the low treatment, they will germinate faster. Okay, so the imbibition time is lower, and the germination rate, the germination percentage is higher. So it is the amount and the availability of the water, it will influence the germination of the seeds. This experiment is showing the relationship between the temperature and also the seed germination. So there are 3 treatments for the temperature. So it's 10 degrees Celsius, 12.5 degrees Celsius, and also 15 degrees Celsius. So from this figure, it shows that the germination rate or germination percentage increases with the rise in temperature, up to the optimum level only. Okay, but if it's getting too hot, then they will exceed the optimum level, and the germination percentage or the germination rate will decrease. So that is how the seed will become the seedling. So let's see on how the shoot is formed. Okay, so the shoot is formed. Okay, before that, let's see. Okay, in one plant, there are the shoot part and also the root part. Okay, so we're gonna have a look at the shoot first. Nanti later we'll have a look at the root part. On how the shoot is formed. Okay, so it's through the shoot apex. So the shoot apex too, they have a lot of meristematic cells, and as we know, the meristematic cells is a part of the plants ataupun part of the cells yang memang actively growing, actively dividing themselves and elongating themselves. So this is the formation of the SAM. So SAM ni stands for the shoot apical meristem, and kita boleh nampak it involves the genetic control. So for the shoot growth and also the root growth pun juga sebenarnya, and also flower growth pun sebenarnya on the cellular and also molecular basis ataupun level memang dia ada genetic control dia. Okay, so kita boleh nampaklah kat sini the gene expressionnya on how it promotes the formation of the shoot. Okay, through the shoot apical meristem. Okay, so this figure it shows clearly lah on the shoot apical meristem, where this is the stem cells and this is where the rapid cell division. So this is the central tissue of the stem, and jadi inilah yang akan will keluar akan jadi shoot. Okay, so this is the leaf anatomy. Ini adalah refresh baliklah berkenaan dengan leaf anatomy. Alright, so this is the top of the leaf and this is the bottom of the leaf. Okay, so top and bottom ni dia ada cuticle. Cuticle layer serve as a protection juga, and then they have the epidermis layer. So they have the upper epidermis layer and also the lower epidermis layer, and on the bottom part of the leaf they have opening. Okay, they have the opening for the water and also for the gas. Okay, so water ni is for the transpiration lah, daripada from inside going outside for the transpiration process, the water vapor, and kalau gas tu is for the photosynthesis. Photosynthesis process where carbon dioxide will go from outside into inside and oxygen will go from inside into outside. Okay, so that is how the shoot is formed by the genetic control. Yeah. Next, let's have a look at the how the formation of the root. Okay, so before that, refresh sikit lah root system ni. Dia mainly we have the taproot and also the fibrous root. Maknanya akar tunjang and also akar serabut lah. And kalau taproot tu dia punya ciri-ciri dia punya kriteria ataupun karakteristik dia, they have one main root, tunjang satu je, and then dia ada a few branch roots. For fibrous root system ni dia semuanya a mass of similarly sized roots. So semua nak dekat sama je dia akar serabut kan. So semua dia tak ada yang taproot yang tunjang. So the function of the root ni there are few. The function of the root, so for example, is for anchor, is for the starch storage, and also is for the absorption of the minerals and nutrient, and also water. Okay, so for root system development ni, so there are four phases atau four zones, four developmental zones lah dekat bahagian root ni yang it will help in the elongation of the root. Okay, so the first one is dekat area dekat root cap ni. Okay, so root cap ni dia ada cap dekat situ. It won't be exposed. So dekat sinilah yang akan protect the root apical meristem, and also it will push down, okay, untuk dia lagi masukkan ke dalam. And then the second zone is the meristematic zone. Okay, so meristematic zone lah dekat sini lah. This is the maximum rate of the cell division. This is very active, so it will push the root cap to elongate. And then behind the meristematic zone tu dia ada elongation zone. So elongation zone dekat sini lah juga dia akan elongate dia punya cells dia, and then it is being helped by the division rate dekat meristematic zone tu, and then it will push through the root cap. And the last one, so this is the maturation zone. Okay, the maturation zone ni, because this is the part yang dia start awal-awal. Because yang awal-awal lah. Because yang baru-baru ni, yang the newly formed cells ni for the root apical meristem, semua akan ditolak ke bawah, ke bawah, ke bawah, ke bawah. So whatever yang dia tertolak ke belakang tu, so that is the matured, ataupun yang lama-lama lah, yang veterannya sini-sini. Yang junior-junior semua dekat bawah ni. Okay. Root hair dia akan ada area maturation zone. So dia tak akan berada dekat bawah. Because once they are mature, dia dah jadi senior, then the root hair will develop. Okay, and if they have the lateral root pun, lateral root and root hair ni, they are different. So kalau lateral root ni, they won't emerge ataupun they won't grow in these four developmental zones. So they will form in the non-growing region. Okay, so that they won't interfere with the elongation and division process of the root. So this is the process where it shows the development of the root hair dekat maturation punya zone. Okay, so this is the first step, second step, third step, and fourth step. So the first step is the cell specification, and the cell is it comes from the individual epidermal cell. Okay, so kita boleh nampak there is a new root hair going out lah. Then the second one is the root hair initiation. Okay, so once it has this, the cell is being specialized to do the root hair, then it will be initiated. Okay, to proceed with the root hair development. Then once it has been initiated, it has been developed, then dia akan ada tip growth. So because the tip tu, remember kan from previous slide, tip growth tu is for protection as well. And last one is the maturation process, because the initiation is complete, elongation is complete, tip growth pun dah ada. So maturation lah, dia just maintainkan saja the root hair growth and development. Okay, so that is how the root is formed. And lastly, let's have a look at how the flower is formed. So this is the flower structure, and the important part is in one flower too, they must have the male part and also the female part. So the male part is called the stamen, where it has the anther and also the filament, and couple as well, where they have the stigma and also style yang dekat bahagian tube tu, and also the ovary. Okay, so stigma ni, it is designed, created so that it is a bit sticky, so that whatever yang enter ni, dia releasekan dia punya pollen, dia releasekan dia punya egg dia semua, so it will be stick through the stigma tu, and then it will go down into the style, and it will meet in the ovule, and so this is where the fertilization process occurs. So there are few signals for flower evocation. So flower evocation, it means that we are telling the plant to produce flowers. Okay, to produce flowers. So there are the internal and external factors. So internal factors ni phase change lah. So maknanya it's no longer in the seedlings punya phase, it's in the maturity punya phase. And then hormones juga. Hormones, it plays a great deal in the production of the flowers. External factors from the environment jugalah, kan. Alright, so light, temperature, total light radiation, and also water availability, memang it plays a great role in flower evocation or flower development. Okay, so as I mentioned just now, because this is the cellular basis kan of the plant growth and development, so of course there is a genetic control on the development of the shoot, the development of the root, and tak ketinggalan lah the development of the flower. Okay, so this is A ni, it shows the longitudinal section. So kita boleh nampak there is a stamen, carpel, petal, sepal, semualah. Okay, so this is in cross section ni daripada atas yang kita boleh nampak. Okay, so sepals is the one dekat luar ni, and then petals the one dekat dalam, and then stamens, the blue color, and carpels dekat tengah-tengah ni lah, because carpels kan dia dekat tengah sekali. Okay, the reason why it shows because all of that, semua yang sepal, petals, stamens, and also carpels tu, they are being controlled by the gene. Okay, so the correct genes have to be expressed at the correct time, at the correct amount, so that all those structures of the flower tu will develop properly. Okay. However, kalau macam for example there are mutations in the floral gene kan, jadi dia akan jadi pelik lah. Okay, so for example this is all the genes required. So they have the A, B, C and E genes required to produce a normal flower. But for example kalau this one ni dia tak ada genes yang untuk A, kan, so tengok flower dia dah different. Kalau yang ni genes untuk B tak ada, flower dia pun it will turn out different. Kalau this one A, B, E and C tak ada, so flower turn out different. And this one lagi lah, semua genes for the flower development tu semua tak ada, jadi memang dah tak ada rupa flower dah. Okay, so mutations do happen. Okay, ada banyaklah faktor for the mutations, but the main point here is that the cellular and also the molecular level, the gene tu, it controls the production of the shoot, production of the root, and also production of the flower. Okay, so again, a refresh on the plant life cycle. So the first one is how the seed is formed. Okay, so the seed is formed is through the fertilization process. So fertilization process tu maksudnya they must have a male and also they must have a female. Okay, so for plants tu, in one plant tu dia memang ada male and also female. So kalau male tu dia punya reproductive cells tu is coming from the sperm cells. Kalau for female tu dia punya reproductive cells tu it comes from the egg. Okay, and then once the fertilization tu dah occur, then they form the seed, kan? So seed tu, remember lah, they have three structures, kan? Basic structures. They have the seed coat on the outside, and then they have the embryo, which is the tiny plant, the baby plant, and also they have the endosperm. Okay, inside the seeds tu, because the endosperm tu it provides the mineral and also nutrient. Okay, for the baby plant. And then the second step is once the seed is formed, then how does it become a seedling? Okay, so this is where through the germination process. So the germination process tu, it means that from the seed tu dia akan break out, dia akan keluar lah the shoot and also the root. So how to um, tell the seeds that okay, it's now your turn, break out and then germinate into a seedling. So there are a few things, the water uptake. Okay, remember water uptake too is through the imbibition process. So imbibition process there is the absorption of water through the by the seeds, and then it will become swelling. So rehydrated cell expansion happen, and then the seeds will no longer be in the dormancy punya period. And then we did see the experiment on the water relation and also the temperature relation. Okay, so we can see that if the seeds are being treated with high water availability, then it will germinate faster. And also if the seeds are treated with higher temperature, but not so high, then the germination percentage is also quicker and faster. Okay, so once it has become a seedling, then let's see how the shoot is formed. Is through the shoot apical meristem, and also the root is formed through the root apical meristem, and both of these are controlled by the gene as well. And for root tu dia ada 4 developmental zones lah kan. So it starts with the root tip tu as a protection, and then dia ada meristematic zone tu. Okay, so that is where the meristematic cells tu are actively dividing, and then there are the elongation zone, that is where the cells are elongating, dia panjangkan lagi root tu, and last one is the maturation zone. Okay, and lastly is how the flower is formed. So the flower is formed too because of the signals from the internal signals and also the external signals, and they're also being controlled by the gene. Okay, so genetic control. So if there are mutations in the gene kan, that control the flower, control the root or control the shoot, then the development of the structure too will be impaired. Okay, alright, so that's all for part 1. We will continue with part 2.